Hello everyone, my name is Jianzhong. I'm from Hainan University. It's my great pleasure to share my most recent research progress with you. Today, my topic is supply-controlled calcium carbonate dissolution decouples the seasonal dissolved oxygen and pH minima in Chesapeake Bay. This work includes many cooperation and efforts from multiple institutions and has been published on limnology and oceanography in September last year. I'd like to introduce my work in the following outline. First, I will introduce the background information about the difference between ocean acidification and coastal acidification, as well as how calcium carbonate cycling interacts with coastal acidification. Second, I will talk about the motivation of this study. Third, I will introduce the materials and methods that we use to approach the research goal. Next, go to the main results and discussion parts. Finally, we will make some conclusions. Human activities have increased the atmospheric CO2 concentration from 280 ppm in the pre-industrial era to beyond 410 ppm at present. As a major sink of CO2, ocean has absorbed about 24% of the total anthropogenic CO2 emissions over the past 6 decades, and the oceanic uptake rate has 2.5-fold increase from 1960s to the past decade. That leads to the increase of surface seawater PCO2 and the declines of seawater pH and saturation state of calcium carbonate. This process is well known as ocean acidification, which is harmful to many marine calcifiers and their habitats. Compared with the anthropogenic CO2 caused open ocean acidification, coastal acidification has more intense variability, which can be one order of magnitude higher, and it has many controls than a classic ocean acidification. For estuarine coastal waters, there are many material exchange among the boundaries. Ocean acidification signal can be delivered into estuaries by freshwater and seawater mixing. Also, the upwelled CO2-rich deep water can decrease pH further. The carbonate contents and species of river input and natural export from the blue carbon systems can have a significant impact to the coastal region. The benthic efflux at the sediment-water interface, the atmospheric CO2 uptake, and wet or dry acid deposition at the air-water interface could possibly enhance coastal acidification. Also, internal activities such as photosynthesis and respiration, calcium carbonate formation and dissolution can affect the extent of coastal acidification. Overall, the controlling mechanism of coastal acidification is very complex, and my research interest is focused on how calcium carbonate cycling regulates the coastal acidification. In June 2020, we have published a paper about a self-regulated pH buffer mechanism in the Chesapeake Bay. Calcium carbonate solids produced within the recovered submerged aquatic vegetation beds in upper bay and shallow waters could be focused into the hypoxic bottom waters in the mid and lower bay for further dissolution, which increased the alkalinity and pH and alleviated the coastal acidification. However, this paper is based on one single cruise in August 2016. We have no idea if this pH buffer mechanism also works in other seasons. So the motivation of this work is to reveal the seasonal distributions of carbonate system, especially on calcium carbonate cycling, to identify their major controls and to quantify their effects on pH dynamics. Chesapeake Bay is the largest estuary in the eastern United States. It suffers from long-term eutrophication and hypoxia and anoxia. We conducted 4 cruises along the main channel of Chesapeake Bay in May, June, August, and October of 2016. The general methods include two end-member mixing and mass balance of DIC and TA, and chemical proxies and stoichiometry. And we also simulate the pH dynamics by CO2-cis. This figure shows the profile of DO and pH from the upper bay to lower bay. We can see the mid-bay subsurface DO is lower than the surface DO in May and reaches the threshold of the hypoxia in June, and continue to decrease and form one anoxic zone in the center of hypoxia in August. And the water column was reoxygenated in October due to the temperature decrease and stronger physical mixing. However, the bottom pH reaches its minimum in June and increased in August. So the DO and pH minima appeared in different time and they have seasonal mismatch. Furthermore, we download the 30 years monitoring data of bottom DO and pH from the Chesapeake Bay program. The y-axis shows the monitoring size from upper bay to lower bay, and x-axis shows the months. We can see the DO minimum appeared in July and August in the mid-bay, while the pH minimum occurred in May and June at the intersection of upper bay and mid-bay. Taken together, the mismatch of the seasonal DO minimum in August and the seasonal pH minimum in June in mid-bay bottom water was not unique in 2016, but was a persistent summer phenomenon in Chesapeake Bay during the past 3 decades. Then we try to figure out the linkages between carbonate system and seasonal mismatch of DO and pH in the Chesapeake Bay. This figure shows the concentration of DIC and TA against the salinity, and DIC and TA increased as salinity increased, but the pattern is not linear to salinity. That means some biogeochemical processes have changed the DIC and TA. First, we used a regional 2 end-member mixing scheme between estuarine end-member CB2.2 and offshore seawater end-member L1 to derive the total non-conservative DIC and TA, which is defined as the difference between the measured values and the mixing line. The total non-conservative DIC and TA were generally positive. That means some process added DIC and TA to the water column. The additions of DIC and TA reached its peak in the mid-bay, then decreased to nearly 0 in the lower bay, and the peak value increased from May to June to August, and decreased substantially in October. Next, we try to decompose the total non-conservative concentrations into 3 components based on mass balance of DIC and TA. LU is used to quantify the fraction changed by aerobic respiration. Delta calcium ion is used to quantify the contribution from carbonate dissolution, and the residual term is attributed to other processes, which mainly include anaerobic respiration in the sediment and air-water exchange. This figure shows the contributions from different processes to the total changes of DIC and TA. For DIC changes, contribution from aerobic respiration increased from May to August and decreased in October. Carbonate dissolution is minor in May and June, but becomes substantial in August and decreased in October. Others increased from May to June, then decreased afterwards. For TA changes, carbonate dissolution is the most important process contributing to TA addition, reaching a peak value in August. Other processes played a secondary role in tear dynamics. Aerobic respiration had a limited effect on tear removal. We also simulated the pH along salinity gradient to see to what extent different processes affect pH dynamics. This table shows why DO and pH minima had a seasonal mismatch. The data are averaged value within the hypoxic zone. We can see mixing and aerobic respiration induced nearly equal pH reductions in June and August, but significant carbonate dissolution increased the pH by 0.71 in August, but very minor pH increase in June. The overall sum of all process to pH reduction is 0.66 in June and 0.39 in August. Therefore, the mismatch of a seasonal DO minima and pH minima can be attributed to the increased carbonate dissolution in August. One may raise up a question at why carbonate dissolution was minor in May and June, but became substantial in August. The first possibility is the change of saturation state of calcium carbonate, that is, omega. As we all know, when omega larger than 1, calcium carbonate precipitation is thermodynamically favored. When omega equals 1, carbonate minerals are in equilibrium with the ambient water. When omega less than 1, carbonate dissolution is favored. Is it possible that omega switching from larger than 1 in June to less than 1 in August? This is the pH against omega in the four cruises. We can see that omega of mid-bay subsurface water were all below 1. Therefore, the stronger carbonate dissolution in August was not caused by passing the tipping point of omega switching from larger than 1 to less than 1. That leads us to speculate that the seasonal carbonate dissolution pattern was controlled by the supply from an external source of calcium carbonate solids. We next sought to identify sources of calcium carbonate production. Using a method in our previous paper, we used the removal of calcium ion to trace the process of calcium carbonate formation along the mainstem Chesapeake Bay. We can see the maximum removal usually occurred in the upper bay and increased from May to June to August, and decreased in October. The seasonal pattern of calcium ion removal or calcium carbonate formation in the upper bay generally coincides with the seasonal cycle of above ground biomass of SAV in Chesapeake Bay, which begins to increase in late spring, reaches a growth peak in the late summer, and declines in the fall, making SAV-driven calcium carbonate formation a strong candidate process for calcium carbonate sources in Chesapeake Bay. And our seasonal analysis reinforces the findings of our earlier Nature Geoscience paper that calcium carbonate solids formed in productive low-salinity SAV beds is a potential source of calcium carbonate to support mid-bay dissolution and pH buffering. Finally, we can gain some take-home message, including: first, the bottom DO and pH reaches their minima in August and June, respectively, in mid-Chesapeake Bay. Second, the mismatch of seasonal DO and pH minima in Chesapeake Bay is mainly caused by strong carbonate dissolution in late summer. Third, carbonate dissolution in late summer was not primarily controlled by thermodynamic equilibrium, but was probably controlled by the supply from external sources of calcium carbonate solids. That's all my presentation today. Thank you for your attention.